Greetings everyone, welcome to Twashta. In this video, we are going to solve this question from CSIR NET December 2019 paper. The question is from section C and it says, analysis of a homotetrameric protein and a double-stranded DNA that had been incubated in standard buffer on native gels revealed that they migrated true to their physical states, tetrameric nature of the protein and double-stranded nature of the DNA. Following hypotheses were made for the effect of adding high salt to the incubation mix and subsequent analysis on native gels. A. The protein would migrate as a homotetramer and DNA in double-stranded form. B. The protein would migrate as a monomer but the DNA in double-stranded form. C. The protein would migrate as a homotetramer but the DNA in single-stranded form. And D. The protein would migrate as a monomer and the DNA in single-stranded form. Then they have asked which of the following combination of hypotheses is most likely. 1. A and B, 2. B and C, 3. C and D, and 4. A and D. Here I encourage you to pause the video and write down what you think the correct answer is in the comments section. Now let's find out if you're right or wrong. Homotetrameric protein and double-stranded DNA was run on a native gel. Please note that it was run on a native gel, which is a non-denaturing gel. No sodium dodecyl sulfate or SDS to denature the biomolecules, and no reducing agent like beta-mercaptoethanol to break the disulfide bonds in proteins. And that is why on a native gel, biomolecules separate in their folded state according to their physical state, that is their shape and size. Then the question is asking that what will be the effect of adding salt to the incubation mix. Incubation mix is nothing but the buffers. So how is the high salt going to affect the double-stranded DNA and the homotetrameric protein? Well, in case of DNA, the phosphate groups of the phosphate deoxyribose backbone are responsible for the negative charge on the surface. And the positively charged ions that will be generated by adding salt will interact with these negative charges and stabilize the double-stranded structure of the DNA. So adding high salt will stabilize the double-stranded DNA and it will migrate in double-stranded form only. Moving on to the effect of high salt on protein. The protein is a homotetramer, that is, it has 4 subunits. Now the interactions that allow these subunits to form a tetramer might be non-covalent, like hydrogen bond, van der Waals forces or ionic interactions, and adding salt might disrupt these interactions as ions generated from the salt will interact with polar groups and charged groups, thereby denaturing the tetramer into monomers. So in this case, the protein would migrate as a monomer. Or the interactions between the subunits might be covalent, like the disulfide bonds between cysteines. Disulfide bonds are strong covalent bonds and they will not be disrupted by ions generated by the salt. So in this case, even after adding high salt, protein would migrate as tetramer only. Now, since it is not mentioned in the question whether the subunits of the proteins are held together by non-covalent interactions or disulfide bond, it can either migrate as tetramer or monomer. And as we have already discussed, the double-stranded DNA will migrate in double-stranded form only. So the correct combination of hypotheses is A. The protein would migrate as a homotetramer and DNA in double-stranded form, and B. The protein would migrate as a monomer but DNA in double-stranded form. That is option 1. I hope you found our explanation helpful, and If you have a question that deserves a video explanation, please let us know in the comments. Thank you.